Okay, so how many different categories of tastant can you detect with those taste receptor cells in the tongue and on the roof of the mouth and on the, the epiglottis that covers the trachea, the sides of the tongue, etc. Well, it turns out that the nervous system utilizes what we call a labeled line kind of system or approach. And this is actually a very beneficial way to sort of organize detection of energy or information. In this case, the tastants, the chemicals that compose our food, and then route through networking into different portions of the brain, to many different areas that allow for tremendous diversity in our perceptual experience of taste, and we'll see what's called flavor. So initially it was, many of us were taught as children, right, that there's, four basic tastes. There's sweet, there's salty, there's bitter, and there is sour, right? And it turns out that there are more labeled lines, like specific, types of taste receptors. There's one very famous one that was identified in Japan that's called umami, which is more like the savoriness, basically, of the food, the glutamate kind of sets it off, MSG sets it off, the sort of savoriness, the meatiness of things that we also have. And recently, scientists have discovered another specific type of taste receptor cell that's responsive to starch. So if you like scones and breads and things like that, there's a special taste receptor, special labeled line, for detection and distribution of starchiness. In addition, there may be one that's selective for lipid, for kind of fat, as well. There's still some debate over that. But one of the reasons why a labeled line system is so useful in some ways is that instead of having separate types of taste receptor cells that basically respond selectively to peach or nectarine or something like that, you actually have fewer genetic instructions to make fewer individual types or categories of taste receptor cell, and then the pattern of activity across those labeled lines can actually be informative in terms of the distribution along networks in the brain that allow for perception of more complex combinations of sweet, salty, umami, sour, bitter, starchy, and potentially fat. So this is a really good organization for this system. Now, interestingly, the sweet taste receptors, right, are actually notoriously difficult to trick. They're very, very good at detecting glucose, sucrose, fructose. They're very specific for the energy substrates our cells need to generate ATP to function and survive. While at the other end of the spectrum, the bitter taste receptors are quite open in what they'll bind. They'll bind many different kinds of chemicals that may be found in food. And this is interesting. The sweet taste receptors, for example, there have been large industrial corporate efforts, right, to try to trick our sweet taste receptors. But it turns out that if if something tastes sweet, if it sets off those receptors but doesn't deliver the food value, well, your perceptual experience of the flavor, of the sweetness, may change with experience, and you may end up not particularly, it tastes a little off, like that the artificial sweetener, right? What's interesting is that sugar is so important. So this receptor, if we could be easily tricked, we wouldn't get the energy molecules that we require. So this is pretty specific. Well, the bitter ones, for example, are kind of a signal that this is something new or something we don't know what this is, and so initially it can give a kind of an adverse sort of kind of response in us. Now,